So what am I going to talk about today? Talking about microbial interactions with humans. And we start off by looking at this pyramid here of interactions. Now, most of you, when you think about bacteria, when you think about things like E. coli, you immediately think about them as pathogens. But in fact, this is a very misleading view, because most of the microbial world doesn't do us any harm at all, doesn't contribute to illness. It is just out there, and in some cases it's actually doing us great benefits. So we have this normal microbiota, as it's called, that covers our body surfaces. We're colonized by that. And if we look at even those organisms that we might say are pathogenic, very few of them are always pathogenic. We often use this phrase, potentially pathogenic now, because we like to hedge our bets. If we see an organism in a sample from a patient, we don't always know whether it's causing disease. We know that it might be associated with infection in some case series in the past, but it's very difficult to know in that individual patient whether it is or not. And as I say, most microorganisms are out there are never pathogenic. There are billions and billions of bacteria in soil. And babies sometimes eat soil and don't do themselves any harm at all. None of those bacteria in the soil are going to do them any damage. So what I'm going to do is take you through these issues, the human-microbe interactions, the human microbiota, look at infections and pathogens, definitions of terms, look at some of the risk factors that lay people open to infection, very brief sketch map of how infection comes about, the so-called pathogenesis of infection and the virulence factors involved there, and just briefly name drop some of the syndromes that we see in the field of infection, so that you're just getting used to the vocabulary as we go along. So let's start with human microbiota. So each of us is carrying this very rich diversity of bacteria. In a sense, we're like a coral reef. We're just teeming with life, and not just in terms of quantity, although there are more bacterial cells associated with any one of you than there are traditional human cells that you learnt about in cell biology, but there are also many, many hundreds, probably thousands of different species of bacteria associated with your body at any time. So this in a way changes our thinking. As microbiologists, we don't like to think about humans as all those human cells and muscle and bone. Basically, humans are superorganisms that are built from all that human stuff, but the microbial cells are equally important. And Julian Davis, an eminent microbiologist, put it this way. He says that humans are effectively large, highly complex microbial communities attached to some relatively uninteresting organic matter. So that's our perspective there. Some people have even suggested that we should consider this microbiota as a kind of multicellular organ in complexity, in terms of size, in terms of the mass of it, probably comparable to the liver. And it's also important that all those bacteria associated with you and some of the other things, the fungi and whatever, they are forming a third genome. So each of you will have heard about the human genome. And usually people talk about that. They're talking about the nuclear genome. There's also the mitochondrial genome. And then there's this third genome where there's at least 100 times as many genes associated with you in those bacteria as there are actually in your quote-unquote human genome. And this little logo here I put up is from an effort kickstarted in the United States a few years ago called the Human-Microbiome project. And this started as a spin-out from the Human Genome Project, saying, well, the Human Genome Project was all very well, but it's kind of incomplete until we have the genomes of all those other organisms associated with us, all these microorganisms that are associated with us. Until we have those enumerated, catalogued, their genome sequence we haven't got the full picture of what it is to be human. To give you some, I'm going to sprinkle in definitions of terms as we go along. So here's one that we introduce now, the idea of a commensal. It comes from the Latin, means sitting at the same table. This is an organism that lives with us on external bodily surfaces. Now, when I'm talking about external surfaces, I have to just qualify things because it might be counterintuitive what we mean by that. So if we take this here, this is a view of what you look like topologically, okay? So you're just this blob, and you've got this gastrointestinal tract running from your mouth to your anus. But in effect, all of the stuff that's in the lumen of the gastrointestinal tract is outside of your body in this topological sense. So that's what we mean when we say these organisms are on external body surfaces. Although they're in your bowel, they're actually topologically outside of your tissues. And the same kind of arguments can apply in other settings as well. For example, in the genitourinary tract, or in the nose and in the respiratory tract. So we have this huge microbiota. Now, sometimes you might see in some of the textbooks the term normal flora to describe the human microbiota. This is a hangover from the days when people used to classify bacteria as plants 50, 70 years ago, and the textbooks and people's jargon somehow hasn't been updated. But we now, if we're going to be precise, we talk about the human microbiota, not only because we are covering other things apart from bacteria, and bacteria aren't plants, but we're covering protozoa, fungi, archaea, metazoa, viruses, all fit into that microbiota. Now, there's been a lot of interest in the last 5-10 years about trying to define the human microbiota, the human microbiome, the collection of genes that go with that microbiota. There was this idea that there would be this core microbiome that would be in all of us. We'd all have something that's fairly similar. In fact, it turns out that that's not the case, and that there is a huge degree of variation from one individual to another. And if you look at a given individual, their own microbiota will be fairly stable. Things sometimes come and go, but for any individual, their core microbiota will stay stable, but it will vary from individual to individual. In fact, it's quite hard to in some ways, to dislodge the components of the normal microbiota. So if I fed you just a commensal E. coli strain, probably wouldn't establish itself in your gut because you already carry, almost everyone, 99% of the people in here will have E. coli in your gut already, and the E. coli that's already there kind of has squatters' rights. It's very hard to dislodge that organism without doing rather drastic things like giving antibiotics and so forth. So this microbiota, I mean, you've seen them in the adverts described as the friendly bacteria, and they do have tremendous benefits for us. One of the things that they provide is what I just mentioned, this idea of colonization resistance. So they kind of have squatters' rights. They are coating all these surfaces, they're occupying all these niches all over us, and in a sense they're competing for space and nutrients with pathogens, and they're preventing the pathogens from getting a foothold. And many bacteria produce things called bacteriocins, which are toxins active against other bacteria. So if an E. coli has already got possession of a particular niche, and another E. coli comes in, it will also be firing out these bacteriocins, which are very likely to prevent the incomer from establishing itself. We know that this gut microbiota is actually important for development of the gut. It influences the epithelium and the endocrine cell development in the gut, production of mucin, and most importantly, perhaps, the development and maintenance of the immune system surrounding the gut and elsewhere surrounding mucosal surfaces. how do we know that? You can actually, in mice, you can actually rear mice that are completely germ-free. And when you do that, what you see is that they have this abnormal development of the gut. So they get very long villi, they get cecum enlarges, they get altered gut motility. And the lymphoid structures, the secondary lymphoid structures that form part of the lymphoid, the gut-associated lymphoid tissues, they don't form properly either. So we know that the gut is, the microbiota in the gut at least, and probably elsewhere, is playing a key role in normal development. The microbiota is also involved in the assimilation of nutrients and production of nutrients for us. So in a sense, there's this complex food web, an anaerobic food web going on in your gut, particularly in the colon. The stuff goes through the small intestine, you absorb lots and lots of nutrients, but what comes through into the large bowel is feeding this complex food web. That microbiota is synthesizing some useful substances there, some vitamins like folic acid, biotin, vitamin K, and it's also breaking down complex polysaccharides into sugars. So Bacteroides as a genus, a very abundant organism in the large bowel. This is breaking down all those polysaccharides, and then there's fermentation going on to produce short-chain fatty acids. And those can then be absorbed, and we can get the nutrient value from those short-chain fatty acids. And this can account for up to for 10% or more of the calorific value of the food that you're taking in being available, because bacteria have helped you break it down. And these short-chain fatty acids now clearly they're thought to have a role in protecting you against cancer. There's also a dialogue with the immune system through these short-chain fatty acids. So there's this whole complex dialogue going on, an interaction going on between that microbiota and your immune system and your gut. Most of the time, it's good for us, this microbiota, but sometimes it does cause problems. And in fact, there's a growing body of evidence over the last 10 years or so to show that it's not just infections, and I'll say more about this microbiota causing infections in a minute, but there are other conditions which you wouldn't normally think of as that being an infectious thing or something to do with microbes, that actually have now been fingered as actually influenced by this microbiota. One example is the so-called hygiene hypothesis, so the idea that we actually are altering our environment. We're living in very clean environments, we're not coming into contact with a wide range of organisms, and we don't have intestinal worms like we used to. You can argue, probably could make an argument that actually in some situations you can look upon intestinal worms and parasites as part of the normal microbiota. And what this hypothesis suggests is that the reason we have a rise in allergic conditions such as asthma is because we're not seeing those organisms, the wide range of organisms that we would have seen in our evolutionary past. The gut microbiota is also implicated in the pathogenesis of cancer, bowel cancer in particular, inflammatory bowel disease, irritable bowel syndrome, diabetes. In all these cases, there appears to be a role. And perhaps most surprisingly of all, there is some evidence to suggest that the constitution of the gut microbiota can have an effect on body weight and particularly predispose you to either being lean or to being obese. And we know this, there's a guy, Jeff Gordon, who's kind of been the pioneer in this field. We know this because you can look at obese and lean individuals and you can see that there are differences in the composition of their microbiotas. Now, you can't say that's causal. It could be that just being lean, not eating much, changes the diet. If you eat 10 McDonald's a day, it's not surprising that your microbiota might be different if you eat just muesli and fruit. But if you take animal experiments, you can take obese mice and transfer their microbiota to lean mice. So that you can make the mice eat the shit of the obese mice, and they will end up gaining excess weight. So here's this experiment. This is a paper. I think it's important even in the first year that you actually get some experience of the fact that knowledge comes out of papers, not just out of textbooks. Here's a paper from a few years ago where they did that, where they said an obesity-associated gut microbiome with increased capacity for energy harvest. So that they, first of all, looked at individuals, they looked at mice as well, showed that there were these differences there, and then they said, furthermore, this trait is transmissible. Colonization of germ-free mice with obese microbiota results in a significantly greater increase in total body fat than colonization with a lean microbiota. So it does suggest that there is a role here. So a lively area of investigation at the moment. We don't understand all of this at all. What else about the microbiota and disease? Well, the microbiota actually is involved in causing infection locally. So Streptococcus mutans in the mouth, if you don't eat refined sugar, it's just part of your normal microbiota. If you eat refined sugar, it ferments that sugar and produces acid and rots your teeth in the form of caries. Streptococcus pneumoniae, this lives in the upper airways, in your nose, throat, and so forth. But if it manages to spread and gets into your into your middle ear or into your sinuses, then it can actually cause infection. Elsewhere, we see the microbiota escaping from these topological confines that I mentioned. So, as I say, what's inside your gut isn't really inside you until you burst an appendix, let's say. In which case, all those organisms that are living inside your gut, in harmony with you, are suddenly inside your peritoneum and actually causing a rip-roaring inflammatory response and actually threatening your life. Similarly, we see stuff crawling up from the gut lumen up into the biliary tract, where it causes these conditions of cholecystitis, inflammation of the gallbladder, and cholangitis, where the whole biliary tree gets infected. Urinary tract infection is usually caused by organisms that are part of your normal microbiota. So normally it's caused by, the most common cause is E. coli, and that E. coli lives in the bowel, and just occasionally it gets out into the perineal area, that region around the anus, and it then can get introduced into the urinary tract. It's an unfortunate fact of anatomy that in females the urethra is so short, so if an E. coli is on the outside, it doesn't have far to go to get inside the bladder and then cause disease. In men, much less common because the urethra is so long. In addition to those kind of situations, probably some of you in this room have had experience of urinary tract infection. We do see the human microbiota causing disease in the hospital environment, in healthcare-associated settings. So Staphylococcus epidermidis is a very common inhabitant of your skin. It doesn't do you any harm. And in fact, even if you injected it into your blood, it wouldn't do you any harm if you're a normal individual. But if we put in bits of plastic or bits of metal into you, then it can settle down on those surfaces and form what we call a biofilm. If we put in an artificial heart valve, this organism can grow all over it and then actually damage the valve, make it become less secure in its bedding down into the normal tissues and so forth, and actually then cause fatal failure in that setting. Now, I mentioned just briefly before about there are alterations in the human microbiota. Let's just revisit that. So we have developmental changes. So the neonate, newborn baby, as it passes through the birth canal, is obviously coming into contact with the microbiota of the mother, and this is where it establishes its own microbiota to begin with. There are changes also that occur later in life, at puberty. The microbiota of the human vagina changes as the hormonal balance changes and so forth, and you end up getting a particular predominance of organisms called lactobacilli in that environment. When we give antibiotics, this is the equivalent of going out and spraying Agent Orange over Southeast Asia. You're effectively deforesting the normal microbiota, destroying almost everything that's there. And when you do this, you get strange things kind of overgrow. Candida albicans is a yeast-like fungus that when you give people antibiotics, this quite commonly will grow and start forming plaques and causing disease, particularly in the mouth and in the vagina. It goes by the common name of thrush. And this, it's sitting there in your normal microbiota, particularly in the bowel, in relatively small amounts normally. Once you give antibiotics, presumably, I don't think it's really quite well known exactly what's happening, but you're obviously opening up some new niches for this organism that would otherwise have been occupied by the normal microbiota. Another organism you may have heard about is Clostridium difficile, and this also does a similar thing. If you give a patient antibiotics, you wipe out all their 90+% of their normal microbiota in the bowel, and this organism somehow in that new environment is able to thrive. And from being a very, very small component of the bowel microbiota and not really doing any harm at all, it suddenly grows to quite large proportions and starts releasing toxins which damage the bowel and actually can cause life-threatening colitis. The other problem we have in hospitals is that people get colonized with bacteria that could potentially cause disease in them or in others, but aren't actually obviously doing so most of the time. So you've heard of MRSA. MRSA is a fearsome pathogen. If it gets into your bloodstream or into a wound, it can cause a great deal of problem. But probably 9 patients who have MRSA are not actually suffering from any consequences of it. It's only 1 that gets an infection. 9 out of the 10 are actually just colonized. And in effect, the MRSA has become part of their normal microbiota. And this means that it's very hard to dislodge it. As I say, it's quite hard to change someone's microbiota deliberately. And so this is an issue, really trying to get rid of this organism. Nearly finished with the microbiota. There's one other problem we have to think about when we're dealing with this, which is that if we're in a microbiology laboratory, we have problems coming from that microbiota actually making it harder for us to achieve a diagnosis of infection. So if we've got someone who we think might have pneumonia, and we say, cough in this pot, and then we try and look at what's in there, we're not going to get no growth. We are going to get a rich growth of organisms, and this may hinder our ability to identify the one that's causing, that is a pathogen. We see this quite commonly well, there's 2 settings here. One is if we take blood, because we want to culture it. So we think someone's got what we call a septicemia, where they've got bacteria in their blood circulating. Stick a needle into their vein, pull out some blood, and put it into some rich broth, what we call a blood culture medium, and we grow it up. The trouble is that the skin itself is teeming with organisms. You stick your needle through the skin, if you're not careful, there's a good chance that you'll sample some of those skin organisms. Now, a lot of the time we can say, well, if those skin organisms, things like coagulase-negative staphylococci, like staphylococcus, they're not going to be pathogenic. If someone's got meningitis, let's say, we can disregard them. But increasingly in hospital practice, we see patients who have complex devices embedded in their tissues, and those devices may well be colonized with coagulase-negative staphylococci. And so when we grow one of these organisms in blood cultures, it can be very difficult to decide whether that's just skin contamination from the normal microbiota of the skin, or whether it's genuinely coming from the bloodstream and represents a deep infection somewhere. Similarly, when we take a urine sample, if we ask anyone to just pee in a pot, what they're doing is effectively, it's like imagining a fire hose. It's just washing away all of the organisms around the opening of the urethra, all the skin scales and all the microorganisms that are there. They're getting washed off into that urine. And if we just took that urine as it was, we would end up with all sorts of things growing, but it wouldn't tell us whether anything was actually growing in the bladder, which is where we want to investigate. And so what we do is we take what we call a midstream specimen of urine. So we avoid taking that very first rush of urine, and then we intercept the stream after a while, after that first rush. And then we have a much better chance then of sampling what's in the bladder. Even that's not quite enough though, because sometimes even then you still get small numbers of organisms coming through from the skin and the periurethral area. And so what we also do is we quantify the growth. We count the number of bacteria. So if we see a large number of bacteria in urine, lots of E. coli, we can say that is likely to be from the bladder. If we see only a few, we can say, well, that's probably contamination from the skin. But this all goes to show that the microbiota here can be a kind of annoying nuisance when you're trying to get a clear-cut diagnosis. Now, this picture here, I did in the previous lecture have all sorts of pictures about the microbiota all over the body, but I don't want to blind you with science. So this is the only one I'm going to put up. Basically just shows you in these coloured drawing, these pie charts here, that it varies around the body. So in some parts of the body, say in the gut, we will have one kind of set of organisms. In other areas of the body, we'll have others. And this is actually just looking at the skin surfaces in this particular picture here. But even there is considerable variation from sort of your forehead down to your toe web in the kind of organisms you're carrying. Let's just take one example. Here we have the oral cavity, several hundred different kinds of bacteria living inside your mouth. Many of them colonized tooth surfaces. They first attach to these acidic glycoproteins that are deposited there by saliva, and then you get this thick layer of those initial colonizing organisms, and then in fact you get other organisms then sticking on top of those, and you get this complex community forming. And you can witness this every morning when you wake up in the morning, because if you look at your teeth, there is all that plaque growing on them, and it is this complex community. We mentioned already Streptococcus mutans as part of that community actually potentially being a problem. And there are efforts going on at the moment to actually catalogue every organism that ever lived in the mouth. There's a whole database of all the organisms that have been described and their genomes and various molecular barcodes to go with it. Right, let's now just cover a few more definitions as we move quickly on. So, when we talk about pathogens, what do we mean by pathogens? Well, they're effectively microbial parasites. They're organisms that can cause disease. So you've got two alternative kind of definitions there. Microorganisms that can cause disease. We mentioned earlier that we sometimes use the term potential pathogen because we like to hedge our bets. Staphylococcus aureus in a wound, well, it is a potential pathogen. We don't know for certain in many cases. Another term we often use is the term opportunistic pathogen, where this is a pathogen that causes disease only in individuals with abnormal host defences. So anyone in this room, you're not going to get that. If we aerosolise Pseudomonas aeruginosa in here, for example, it wouldn't do you too much harm. But if you had cystic fibrosis, it would then colonise your lungs and could well kill you. Infection. What is infection? Well, this is a situation where a microbe is established. It's obviously got to establish itself on your tissues and then in your tissues. But it doesn't actually have to harm you. It doesn't have to be obvious to you. So we often use the term subclinical infection to describe infections where if we took samples and we looked down the microscope, we did histology on those samples, we could see that there was bacteria. There were bacteria growing there, that maybe there was an inflammatory response or there was some kind of thing going on, but you wouldn't know about it. Probably the most common example of this worldwide is tuberculosis, where you have people, when they get infected with the organism, they have long periods that last many years where they don't realise they're infected. You may see lesions on the chest x-ray, you may not. but there is nonetheless infection going on. The bacteria are slowly growing. They're holding their own in the body, and at a later date they can then break out and actually cause disease. So disease is where we're actually seeing damage to the host, which is crossing a threshold of awareness. You're actually clearly damaging you. You're aware of it. Pathogenicity and virulence, these are terms that are bandied around. Pathogenicity is the ability to cause damage. Virulence is, if you like, a quantitative measure of pathogenicity. A bit of a silly kind of definition, sort of difference in definition there. When we talk about highly virulent organisms, using this as a quantitative measure. The other term that you should come across here on this page is attenuation. You should familiarise yourself. This is where we actually have a pathogen that was virulent, and then its virulence is lost, or at least is decreased because of something or other. So either you've given an antibiotic or a vaccine, or you've made some genetic lesion in the pathogen so that it doesn't cause the normal virulent infection. Okay, I've prattled on a long time now. I'm just going to have a bit of audience participation. Now, Julia Lodge told me that you all know this guy. You've already been told about him. Does anyone recognise who he is? This is Robert Koch, who was one of the pioneers of microbiology, and he came up with these things called Koch's postulates. And I've been told that you all know about this. So I'm not going to tell you what they are. I'm just going to remind you that you should already know that. So if you don't already know that, you need to start thinking, you're getting a bit forgetful. You should have listened in that previous lecture. But basically, Koch had this idea that you have to get things in pure culture and show that they were associated with the organism and so on. But in fact, we now recognise that Koch's view of the world, that single pathogen, single disease, was a little bit simplistic. And if we look at any infection, almost any infection out there, the classical textbook description of the disease is in effect the tip of an iceberg. Beneath the waterline there, beneath the threshold of being obviously that disease, we have all these other things going on which are not quite so severe, but the patient's still unwell, or there may be many infections that are subclinical. So to give you one example, let's take Legionnaire's disease. So this is a kind of pneumonia that you catch from a bacterium. It's caused by a bacterium called Legionella, which lives in water supplies. It's often associated with air conditioning systems in hotels. And when it was first described, it was found at a convention of the American Legion, which is where the name Legionnaire's disease comes from. And it did strike down many of those Legionnaires with a very severe pneumonia, and many of them died. But when people looked more carefully, they realised that actually, for each of those people that got severe pneumonia, there were 2, 3, 4, 5 other people who'd been exposed, but maybe got a bit fluey for a day or two, something called Pontiac fever, but didn't actually get a rip-roaring pneumonia. And then if you go and take blood tests from people and measure whether they've actually made an antibody response to the bacterium, you find a much larger number of people do actually make antibodies. So they've had subclinical infection. They've not actually said, I felt sick that day, but nonetheless they clearly have been exposed to the bacterium. And this is a thing that we see playing out across many different types of infection. Another way of looking at it is that there's a spectrum of virulence. So very, very few infections are 100% clinically obvious. Rabies, if you don't vaccinate, I guess we could argue that you get rabies, you get clinical rabies, that's it, game over. But even there, if we vaccinate, we can change that so that if someone's vaccinated after they've been exposed to rabies, they will go on and not develop rabies. In the middle there, rubella. So in the days before we used to vaccinate everyone, we used to vaccinate young women at puberty. And what you'd find there is if you went and said, have you already had rubella? and then you looked at the antibody test that you might do on the blood from an individual, you'd find that of all those that were positive, had a positive response in the blood saying, yes, they had rubella, only 50% of them would say, well, actually, yes, I know I had German measles. I remember my mum told me, or whatever, it was obvious. And then if we look at another extreme there, poliomyelitis, paralytic poliomyelitis, where you actually get a severe infection that causes paralysis. This is only 1 in 1,000 to 1 in 100 individuals who get exposed to that particular virus and have that virus proliferate in their gut. So what else is there that explains why it's not simple? Like here's a pathogen, causes infection, that's it. There are host risk factors out there. So not everyone is at risk. If we sprayed Legionnaire's disease into this room, some of you would be, well, not this room, but let's say we took a mixed group from the population, went to New Street Station and did it there. Some people would be much more susceptible than others. So people who are compromised in terms of immunocompromised, their resistance mechanisms, those are going to be at risk. Extremes of age, so the very young and the very old, particularly the very old for Legionnaire's disease. Stress and starvation are known to predispose to infection. Disruption of normal barriers, we'll say more about that in a moment, and congenital and acquired immunodeficiency. So in hospitals, when we treat people for cancer, we often make them immunodeficient for a short while, and they become much more susceptible to infection. If someone has AIDS, have HIV infection that's become clinically obvious, again, they are much more susceptible to infection. Now, immunologists will tell you all about the powerful role of the immune system, the key role of the immune system in protecting you from disease. But they're exaggerating a little bit, because the most important thing that protects you is actually the integrity of these physical, chemical, and anatomical barriers to infection. So in having an intact skin surface across the whole of your body is protecting you from infection 24/7. If you have severe burns, let's say, or you have major trauma that breaks open the skin, that is a major predisposition to getting infected. And there are no bacteria which can actually burrow their way through intact skin. And it's the integrity of the... And the same thing applies to the lining of your bowel and of your bladder and all these things. Those intact mucosal surfaces are very important. All of you have this mucociliary escalator pushing out mucus, removing microorganisms from your throat, from your lungs, and so forth. Normal movement through the bowel, peristalsis, is pushing organisms out, as is the normal flow of urine. And of course, almost all of you here are carrying an acid bath around with you in your stomach. All that highly acidic gastric content, you plunge, 99 bacteria out of 100 plunge in there will just die because it's such an inhospitable environment. And you also have things like lysozyme tears. You have certain kinds of immunoglobulin, IgA, associated with secretions on these surfaces. When we go from colonization to infection, what do we mean there? Well, we have this mucosal surface here, some human cells in a row here, coated with mucus in green, and then our microorganism there is shown in blue. So many organisms will just sit on that mucosal surface and not do any harm at all. Some of them will sit on that mucosal surface and cause disease actually while still in the mucosa. Some of them, as you can see here, can actually then invade through the mucosa, get into deep tissues, enter the circulation, and cause more widespread disease. What sort of things are important in causing disease? First, colonizing these surfaces and then causing disease. Well, availability of nutrients is very important. And Napoleon once said an army marches on its stomach, and the same is true for microorganisms. One of the things that our body tries to do to prevent microorganisms from living in us is it severely limits the availability of iron. And in fact, if you get a bit fluey and you have this thing called the acute phase response, the levels of iron in your body fluids go down even lower. And so to survive in these environments, anything that causes disease has to be able to scavenge iron very, very efficiently. So the bacteria can grow locally at the site of infection or spread throughout the body, and they can use what we call adhesins, allow them to stick down, toxins, invasiveness, and other virulence factors. So adherence is very important. Bacteria can stick to epithelial cells. They can have specific surface receptors on those. There are all sorts of extracellular macromolecules and filamentous organelles that help them do this. They have a thing called slime capsules. They have fimbriae, as they're called. As you can see here, these hair-like structures that allow them to stick down. They produce a range of exotoxins, and again, Julia reassures me that you've already had a little bit of exposure to the idea of exotoxins. So these are proteins outside of the bacterium secreted into the external environment which have enzymatic activity that can damage you in various ways. And we can target these with antitoxin antibodies. We can actually make vaccines from them called toxoid vaccines if we denature them. Here's a very nasty example of a specific kind of exotoxin, exoenzymes, in fact, where the enzymes actually are active in the extracellular space. Here's a patient here with gas gangrene as a result of the production of a toxin known as Clostridium perfringens alpha toxin. Toxins can act inside cells as well as outside cells. Often they have this AB arrangement or AB5 arrangement. They have an active subunit and a binding subunit. Here's an example, diphtheria toxin, where it's an AB toxin, where the active part has this enzymatic activity of being an ADP ribosyl transferase, which we'll say more about in a subsequent lecture. Right, the last 2 minutes now, I just want to, again, as part of providing you with the vocabulary to approach this subject, I'm just going to take a very quick whistle-stop through some syndromes. So some of the kind of syndromes, the infectious diseases that we recognise, we define them by the pathogen causing it, in the kind of way that Koch was talking about. So typhoid fever is caused, as a matter of definition, by Salmonella typhi. Legionnaire's disease by Legionella pneumophila. Tuberculosis by Mycobacterium tuberculosis. Diphtheria by Corynebacterium diphtheriae. But there are many other infections where there's not just one cause, one organism that causes it. We may see many different organisms all kind of converging on the same kind of disease pattern. And there we define the syndrome by site. So we talk about urinary tract infection, pharyngitis, pneumonia, septicemia, and so forth. So just to very quickly, saying less than a few minutes, go through these, just to provide you with some of the terms, because a lot of this is, this early stage of coming to grips with the subject is just mastering the vocabulary. Urinary tract infection, you get infection of the urinary tract. Lower UTI is cystitis, is infection of the bladder. Upper UTI we sometimes call pyelonephritis. This is where the kidney is also involved. And we get these signs and symptoms. Dysuria means painful urination. Frequency means you're passing urine more often than usual, and you may get pain as well. We already mentioned the MSU. Meningitis is infection of the meninges and the adjacent brain, and you get these signs and symptoms there. Some of those you might not be familiar with. The term photophobia means you're scared of light. So the patient with meningitis will often want to sit in a dark room, and they will hate it when you try and shine a light on their face. Malaise is just a fancy medical word for meaning you don't feel very well, just generally feel dreadful. And for this we take a cerebral spinal fluid sample. Septicemia, I mentioned before, this is where you get infection of the blood, where the bacteria are actually multiplying in the blood. In its worst case, we see what we call septic shock, where basically blood pressure drops dramatically and the patient will die if not treated properly. And to diagnose this, we take these kind of bottles here called blood culture bottles. They're very rich broth in those bottles. One of them has an aerobic atmosphere, one an anaerobic atmosphere. We put some blood into each of those, and that helps us get a diagnosis. This rather grisly picture here is of an infection of the heart valves, all chewed up. And this is Not projecting in the same colours on the screen there as it is down here. Endocarditis is infection of the heart valves. We get fever, malaise, weight loss, heart failure, and again we take blood cultures for that. Osteomyelitis is where we get infection of the bone, and you can, if looking at this X-ray here, you can see all this broken bone with bits of the bone actually starting to become radiolucent because they've got infection in them there. Gastroenteritis, probably fairly obvious what that is. Pneumonia, infection of the lungs. Again, one of those jargon terms, dyspnea, means difficulty in breathing, or shortness of breath. Pharyngitis, infection of the pharynx of the throat. And that's me finished. So I've taken you through human-microbe interactions. We've looked at what the human microbiota does in health and disease, looked at infections and pathogens, definitions of terms, risk factors and defences, and we've just briefly sketched in pathogenesis and virulence factors and syndromes.